third and the final speaker for this morning is Wei-Zen. Dr. Wei-Zen received her PhD degree from Brown and then was a postdoctoral fellowship, did a postdoctoral fellowship at University of Connecticut Health Science Center at Framingham, Connecticut. She joined the faculty at Wayne State as an assistant professor. Then she became the associate professor at the Karmanos Cancer Institute. And she has been a professor of oncology at the Karmanos Cancer Institute, Wayne State University. She also holds the Herrick Endowed Chair for Cancer Research. Her lab's interest is in tumor immunology and immunotherapy. She has developed a HER2 genetic vaccine, and it has been tested in multiple strains of mouse. And of interest for us is that she works with domestic cats too. So she has done some work on that. She'll be presenting some of the research from that. And also she has conducted human clinical trials. Her lab or her group was the one who started the MCF7 cell lines that we use in our labs for cancer research. And more importantly, she is very passionate about training graduate students, postdoc, and research fellows. And she's passionate towards that. She has trained a number of students. She is well-funded, and her NIH grant started, one of her NIH grants started in '97, and it still continues. And she's a pioneer in a lot of immunotherapy work in cancer research. So without further delay, I call upon Dr. Wei-Zen to talk about actionable targets for breast cancer intervention identified using diversity outbred mass. Welcome.
Thank you very much for that kind introduction. I believe everybody can hear me just fine, here and online.
Sound good. Thank you.
Thank you. So it's really an honor and privilege to be here and to learn about your work and to have the opportunity to share with you some of our own findings. And the two talks before me were so astounding. I was so captivated, and then it dawned on me, you're up next. Okay, so here I am. And what I'm going to do is to first just give you a very quick, okay, this is happening again. I'm not advancing. Oh, there, thank you, Force. So I will give a quick summary in one page of what I'm going to say, and then you can gauge for yourself the bandwidth you want to dedicate for the next hour. So the story is simple. We started from a simple concept of developing HER2 or oncogene-based DNA vaccine. Use that to prevent cancer in human or in vet animals. And in the process of doing that, we recognize the genetic background greatly impact how you develop immune response, whether you develop immune response. And then we found that also impacts tumor progression. And some of these factors that we identify turns out to be actionable targets that you can use for disease intervention or prevention. So the more unique feature that I'm going to talk about is our use of diversity outbred mice, because each one has a unique genetic composition, and they were designed in such a way that you can track and map which gene actually contributes to the phenotype you are interested in. And so we have a battery of genes that are of interest, and which one would you like to use? And so how do you prioritize and how do you choose? And the molecule that I'll be coming back to a lot is HER2. We use, it's a tyrosine kinase receptor. It's a major oncogene, as you're familiar with. And there is a red homologue called NU. And when you express NU in the mice, they develop spontaneous memory tumors. So I will refer to them as oncogene and immunotherapy target. So this in a nutshell is what I'm going to cover in the next hour. So the first part is our genetic vaccine work. By now, I don't have to defend using genetic vaccine because most of you have already got multiple genetic vaccine in you and you know it can work. So this is light and day from the time 20 some years ago, we first reported HER2 DNA vaccine. It was an uphill battle. And so here is the HER2 that is the transmembrane protein with extracellular intracellular domain. And the kinase domain in the intracellular region is where they transmit signals and cause cancer. So we have 3 series of vaccine we developed over time. The first series was just to remove the kinase activity so you can express the protein as a vaccine without having oncogene concerns. There are various constructs. The simplest way is just to cut it out. So we have extracellular and transmembrane domain. And this first generation vaccine has been used in several trials. The first one done in Karolinska was reported over 10 years ago. Then various pharma company has done it in US and Europe. The most recent one was a vaccine led by Jay Berzofsky, and where they took this particular construct of ours, put it into an adenovirus, and use adenovirus to infect dendritic cells of the patient, and that dendritic cell is the vaccine. So that was reported last year. And I will go into just a little bit more later. And after a while, we of course know this HER2 is a self-protein. And if you want to induce an immune response to self-protein, it's always a little challenging and it doesn't induce a very strong response. So we try various ways to make it immunogenic without losing critical epitope. So one way is to take a gene from the same gene from a related species, but not identical. So they have just a little bit of difference and using that little bit of difference as an adjuvant. So one of the construct that I will be talking about more is the hybrid. Half of the extracellular domain is from human, the other half is from the rat new, and the sequence identity is about 88%. So the hybrid vaccine works a little, quite a bit better than the native protein. And I should mention that we always inject the DNA into the muscle and use that as a depot, no different than the mRNA vaccine you receive. It's an intramuscular injection. And rather than the nanoparticle you use in the COVID, we use electroporation to shove the DNA into the muscle cells. And so finally, in the 3rd generation vaccine, which is more recent, we figure out a way of rather, instead of putting a whole chunk of foreign HER2 gene, we identify 5 residues we can substitute. And that keeps the HER2 structure, keeps the critical epitopes, but give it enough foreignness so that it induces a pretty good response. So this is the history of our HER2 vaccines. And what did we find when we immunize animals? First, we generate a human HER2 transgenic animal, and they don't develop tumor, but human HER2 is their self-antigen, so they respond to HER2 as humans may be expected to as a self-protein. And this is available from Jackson Lab. Anyone can request for them. And we bred these mice into C57 black background or bulb C background or bulb by B6 background. And we vaccinate them by electro vaccination of this naked DNA. We measure anti-HER2 antibody level. If the mice are of B6 background, they have very little response, usually maybe 3 to 5 microgram per mil of antibody you can detect in the serum. But if they're in bulb background, they respond way better. So it's about, average 30, 40, 50 microgram per mil. It's really quite a striking difference. The student who was doing this as her thesis got quite frustrated when we crossed the mice more and more into B6, she was getting less and less immune response. Fortunately, she prevailed at the end and reported this work. If you you make F1 strain of 2 parental strain, you get intermediate response. And this is when we ask ourselves, does the genetic background really have a profound impact? And is it important? Because people say, everybody knows B6 mice are more Th1, bulb C is more Th2, so what's the big deal? And this question has been sitting there, as you can see, a couple of decades. But is it important or is it not important? Is it worth looking into? Is there something to be harvested? So that's the question that kept hanging in our mind. And if you look at human trials, Dr. Ochoa said, usually 30% response. That's almost like golden. Whatever immunotherapy trial, you often get 30% response. And why the heck? So our vaccine trial, that's really Jay Brzozowski trial in NCI. They put this adeno HER2 vaccine, and you can see a similar plot, tumor growth. And you can see some of the patients have pretty good response. Some of the patients, their tumor progress. And this is a patient 9 who has a gastric cancer. This is all comers that have HER2 positive cancer. The original cancer went away after vaccination, but new cancer arose. So in patient number 7, which is an ovarian cancer patient, which is this one, is a very striking response. Eventually something recurred, but then that tumor lost the HER2 expression. So there is this selection. You lose the antigen, then of course your vaccine response is not of any significance. But what stood out to us is, again, this is quite a variation. Sure, they may have different disease, may have different epigenetic environment, on and on, the list goes. But are there factors that's in the genetic background that predetermine how well you will respond? Is it worth going into? So we thought, humans are very hard to do this kind of study. Mouse may be too artificial. So how about some kind of an outbred animal that actually develops breast cancer? Can we ask this question in that kind of situation? So we decided to look at the response using outbred cats. Why? Because if a vaccine works here, maybe it's a veterinary medicine and can be used to treat our companion animals, and that it would give us further information on whether genetic background or some genetic elements really do regulate the immune response. So, and cats turn out to develop memory tumors. Here I'm speaking in a vet school, and you know better than I do. But 18% of cats that are female breeders, they do develop memory tumors. So this is a true disease in the cat, and these tumors oftentimes are malignant and can cause a serious problem, and they are treated, as far as I know, by mastectomy. So, and many of these tumors express HER2. So we thought we should test this in our own hands. So we collaborate with a local vet referral service, the Oakland Vet Referral Service, and they recruited some patients for us. First, we want to know if HER2 is expressed. We took 2 cat cell lines, K248 and K12. They are both cat memory tumor lines, and we look at the expression of HER2. Yes, they express HER2, and this is the human ovarian cancer control, and that has amplified HER2, so that has a lot more. But indeed, everybody expressed HER2. So they also express EGFR. And then we have these 3 patients. Chanel has 2 tumors, and the other, Cleopatra and Lily, each has 1 tumor. Then we compare this with K248 xenograft. So everybody has HER2 expression by immunohistochemistry. So we say, okay, this is probably a reasonable place to test if HER2 vaccine can be effective in inducing immune response. So which vaccine should we use? So this is cat. If you look at cat HER2 sequence, that is fully sequenced, reported. But we know if you use cat HER2 by itself, it may not be so immunogenic. So we can introduce some small modifications, or we can use HER2 from a different species, which is highly similar. So we have black bear HER2 that we prepared ourselves. And that is 96% identical with the cat HER2. Human is 93% and rat is 88%. So we purchased purpose-bred domestic short-hair cats. These are healthy, beautiful cats. And we vaccinated them. We took very good care of them. And after the experiment, these cats were adopted by the caretaker, and they lived a very happy life thereafter. And we can still hear about them. Apparently, they all grow happily. And so here is just a brief summary of maybe about 10 cats, and we were giving various kinds of vaccines, whether it's feline HER2 with a point mutation, or the hybrid vaccine that combines human HER2 and rat nu, or the bear vaccine, or a combination, Sure, it's a hodgepodge of different vaccines. But if you just look it over, 3 cats in this group have a pretty good response. And this is T cell response measured by interferon gamma production. And again, about 30% response. So these are not patients who have such different background. These are cats bred and kept in a controlled environment. They are happy cats. And yet when you vaccinate them to a HER2, which you try to induce a self response, they vary quite a lot. So by then we say, we're pretty convinced there are some heritable factors that really impact the response. But how do you get to it? You do genomics, proteomics, whatever. How can you get to these genes that is making a difference? So just at that time, I learned about a new mouse model that was in the making for about 2 decades by the mouse geneticists. They want to analyze complex trait, whether it's obesity, whether it's cancer, with whatever it is. They want a system, a mouse model that will allow you to do the genetic mapping, somewhat like human GWAS, but much more precise than human GWAS. So what they did is they took 8 mouse strains, and this color represents different strains. It's cut in stone. So the 5 strains that are mouse laboratory mice are AJ, B6, 129, NOD, and NZO, and they are in this designated color, and they are called A, B, C, D, and E. This never changed. This is cut in stone. But they also took 3 mouse strains that are wild in origin, and they have been inbred into syngeneic mice. So they are Cas, PWK, and WSB. And why were these 8 strains chosen? Because together they represent more than 90% of all the genetic variations in the mouse species. And most of them are single nucleotide polymorphism. So you have a gene that the gene in you and in me may have a little bit of sequence differences, single nucleotide change, and sometimes that cause the level of expression to change or the structure or the function to change just a little bit. So there were a total of 38 million single nucleotide polymorphism in here, about 7 million insertion, deletion, or structural changes. And you can see this is the number of single nucleotide polymorphism, and 18 million each come from Cas or PWK. They are most different than the other strains, and they often give you the most information because they have more markers. And what they do is they take these strains, cross them, never brother, sister, always to a distant strain. And what happens is that, you have more and more recombination. And at the end of the day, you come with a funnel that has these different combinations of mosaic of chromosomes. And then in Jackson Lab, they keep 144 of these funnels. And then they cross, never brother and sister, always cross outbred. As a result, 3 times a year, you can buy these diversity outbred mice from them, and each mouse would have unique mosaic composition of their chromosome. Here is the 20 chromosome. Each one, you can see different color represent different strain of origin. It never changes. And you can get, in the 8 founder strain, of course, have been fully sequenced, totally understood. And so therefore, you can trace back your individual mouse, have the whole sequence traced accordingly. So if you have a phenotype you want to measure, whether it's obesity, whether it's treatment response, whatever, as long as you have a phenotype that can be reduced to a single number, you can correlate that phenotype with the genetic composition. So what happens then, we want to look at the immune response to HER2 in this particular study, and we take our human HER2 transgenic mice and take the one that's in B6 background. So 2 chromosomes, one of them carry HER2. These are kept as heterozygous because homozygous mice die in utero. So we take this mouse and we cross it with diversity outbred. The F1 mice has all kinds of color and shape, and keep in mind, DO mice have a lot of wild mice origins, so they're quite wild. They can jump straight up 3 feet. They can bite like you wouldn't believe. So you have to be a little bit gutsy to work with them. And so here is the mouse that has 1 chromosome coming from the B6 parent with a HER2 gene in there, and the other chromosome is totally mosaic. So there are 8 possible ways of having haplotype for any particular gene, and this is your tool of mapping. And they were immunized with a clinical vaccine that's used in a trial. Here is the annual HER2 vaccine. We didn't do the DC, we just give the adenovirus directly and measure the HER2 IgG response. So if you take the parental strain of B6 mouse and you immunize them, as I said, the response is poor, maybe 4 or 5 microgram per ml. If you take these mice that has diversity outbreak background, it spreads as wide as you can. So changing the genetic background drastically changed the response of these individual animals. Can you trace them? And this is most prominent when you're looking at immune response to the self-HER2. If this is a mouse that doesn't express HER2, so HER2 is foreign, then you can see the immune response is much more homogeneous. So this is a big challenge, particularly for cancer vaccine, because the genetic background or some heritable factor does impact how you respond to your own tumor. And so what are the tools? First of all, you want to know how to identify the genes in each mouse in order to do the mapping. And so our mouse geneticist friend actually have developed these tools for us, and they have a chip that has 143,000 markers evenly distributed on the 20 chromosomes and encompass the 8 different haplotypes. So you take this tail and you ship to this central location where they do all the genotyping array for you. And it comes back, and it's not so bad, it's just over $100 per sample. And you can, according to the 143 markers, extrapolate back the haplotype of each segment. And that translates to the full sequence because all the parental strands have been mapped. So now you can do linear regression, taking the HER2 antibody level, regress it according to the haplotype at each marker to see which one, which segment, which SNP or which marker location actually has a correlation with either high or low antibody response. And then you can have a value, log of odds, which basically is a p-value on how likely this is true finding rather than an artifact. So the p-value 5 means probability is less than 10 to minus the 5th. So it seems relatively stringent. So with this process, we identified a region in chromosome 17 where the log of odds value is close to 6. And this is each one, this is chromosome 17, and it would have 7,000 markers on them. And each marker has a log of odds p-value. And so let's say, just look at this region, the value is very high, saying the genetic background does impact, and which background renders high or low response. This plot of this whole thing is done, of course, with some algorithm, and it's called RQTL. It's available online free, and the community of the complex trait people are fairly friendly. If you ask them, they'll try to help you, and Jackson Lab will try to help you. And so in here, it says, this is the average tumor onset age, and this is the average antibody titer, and this is high titer. So the one that's high titer here would be yellow or blue or sometimes red, but yellow is AJ, blue is NOD, and red would be PWK. So when these 3 strands, this 3 kind of haplotype background renders high immune response, and it is in this particular chromosome region. So which gene is in there, that you can actually find out, because you can trace down all the genes in single UCSC genome browser, and then you can go to, used to be, Sanger's mouse genome database. And for example, here is a gene that stood out to us in chromosome 17 called H2T23. And in the Sanger database, it tells you these 8 strands, the reference is B6. What is the marker? So there are single nucleotide polymorphism when you compare to the reference phase, and you can see in this particular region, AJ and OD have a lot of unique changes, and that's unique that the two of them share, but unique to everybody else. And every so often you will have some PWK. So yellow means missense mutation, and red is stop codon. So when you have this kind of changes, you probably have some changes in the protein, whether it's expression or function. So, you know, with just this much information, we say H2T23 looks kind of interesting. Why? Because H2T23 is a homologue of the human HLA. We know it binds to NKG2A, which is an NK cell checkpoint molecule. So that if this is triggered, it actually sends negative signal. So if this molecule is important in inducing a HER2 immune response, does that mean NK cells are important? So that when NK cells is shut down, maybe your immune response is not so good. So we said we can do a preliminary validation experiment. So we took our B6 HER2 transgenic mice, we cross it with AJ or we cross it with NOD. Those mediate higher response, at least according to our RQTL algorithm. And so now we look at the antibody response. Now they average about 40, 50 rather than 4 or 5. And the NOD F1 mice is about 10. That's still better than 4 or 5. So just by crossing into those suspect strain, we're getting better immune response. And so is NK cell contributing? So how about depleting NK cell? We use specific antibody to deplete NK cells. And sure enough, you take away the elevated antibody response. So we felt that it's okay to conclude then that NK cells positively regulate HER2 vaccine response in these healthy animals. And there has been clinical trials targeting NKG2A together with other treatment. But maybe it's time to revisit those trials and ask if the trials were set up in a way that would directly ask this kind of question, or should we start a new trial now that genetic vaccine is coming back in fashion, that BioNTech is claiming they are going to have one in 2025. Maybe we can crawl out of our cave and say, okay, now maybe we should look at this seriously, and there's some data that would help us make this work more successful. So it's for this part, so I have two DO-QTL analysis experiment to tell you. This is the first one, and I think it's not too bold to say that these mice can be an effective discovery platform for regulatory genes of complex trait, such as tumor immunity. And we have a good chance of finding actionable targets, such as NKG2A. And of course, the region in chromosome 17 encompasses quite a few other genes, you know, like there's one DO that is important in peptide processing and antigen presentation. So there are many other possibilities. This is just one example, and it's already showing quite significant impact. And so in our group, we feel this tool is really quite profound for those of us who work with a single molecule for 20, 30 years. Now we suddenly have a plate full of molecules, and they have demonstrated in vivo. We pull it out from a live situation, not speculation, but the system tells you this is the molecule. So we said, is antibody response or immune response the only thing that we can draw information from? Tumor immunology involves two parties. There is immune system and there is the tumor. So what's happening with the tumor? Are tumors free or independent of the genetic background? So let's ask. So we decided to look at another platform. We take bulb C mice that express the transforming nu and develop spontaneous memory tumor. We are pretty strong, stuck in the mud about using spontaneous tumor, because the tumor developed in the environment with the matrix, with the immune infiltrates, rather than you implant something in there, in 5 days you have a tumor. That's a very different scenario than when the spontaneous tumor come up. And in that environment, you try to manipulate the immune system. So we just decided to take a very simple measurement. If you ever want to do this, try very simple readout. Any manipulation, you put your human hand on there is a variable. And keep in mind, our readout is variation. So if you introduce variation into your readout, you're going to get in trouble. And the number of mice you need, in the last experiment, we used a total of about 150 mice. And because there are 8 haplotypes, when you use total DO mice, the golden standard is 500 to 700 mice in your as N. And keep in mind, we only use female, we only use transgene positive, so we usually breed 500 mice to select 100 to use. But you'll realize it is important to do that number. So here is bulb new T mice. And if you take a CT scan, because of the new T expression, all 10 mammary glands have palpable tumors. And the liver and kidney is where the tracer goes. So that's non-specific, even though they're very bright. And we can detect as if the new gene, oncogene, is expressed all the time. So we did an immunopet using the copper-64 conjugated antibody to red new, and sure enough, they're taken up by every spontaneous tumor. So this is a model. We feel confident it's developing spontaneous tumor in a natural environment. It's an oncogene that you see in human, and therefore the genes we map may correlate with human breast cancer. I'm not going to put anybody down, but if you use a tumor that is a non-oncogene in humans and you do this kind of assay, you're going to find genes, some of them relevant, some of them is relevant to that antigen you use, and that may not translate to human. Okay, so here is what tumors happen. If you measure in the bulb new T mice, every mouse develops spontaneous tumor at the age of about 14 weeks to just about 20 weeks. Every mouse, 100% penetrance. Sometimes we don't see it. Then we retype, or we say, do we have an infection in the colony that makes the mice not develop tumor? But if you cross them with a different strain, B6, for example, just by crossing, you see the tumor onset is way delayed. All you need is to put a different genetic background, then this story is thrown out. So we believe this is telling us that there are genetic factors that's regulating. And so which gene is it? If we look at the genes in the tumor, we take the mammary tumor from here, we take normal mammary gland from a normal counterpart, or we take tissue that's right adjacent to the mammary gland. You do a single cell RNA-seq and you say there are about 6,000 genes you can measure with confidence. So we say there are genes we suspect that's regulating. So which of the 6,000 is doing the job? Okay, now we've been through this RQTL algorithm once, so right now it should be quite familiar. So we take the bulb new T mice, again, it's female, it's heterozygous, 1 chromosome expressed new. Cross it with DO, so now you have the F1 mice, and these mice, both chromosomes are mosaic, so there are 36 possible diplotypes. These mice have 1 chromosome that's mosaic, so there are 8 diplotype or 8 haplotype. This makes the sample size way more manageable. We can get meaningful data for 86 mice. If we use this, we'll need 500 mice. So using F1 mice, we take these mice and we look at the tumor onset. Here is the bulb new T mice, again, 14 to 20 weeks, like clockwork. These mice, DO mice, don't make tumors. If you take the F1 mice, now the tumors begin to push forward rather than 14 weeks. You begin to see them when they are 9 weeks of age, and they grow and they spread out. They don't delay much, they just spread out. So if we summarize this data, the open circle is the bulb new T mice. This is the age when we first detect tumor, and they peak at about 16 to 17 weeks. If you do the DO mice, they peak at about 12 weeks, 12, 13 weeks. So there are some things, some molecules, some genes that's pushing the tumor to go early, to be more aggressive. Therefore, is it possible to find these genes? And I hope you are now more inclined to think that there may be. So we take the tumor onset age, which is a single number, do linear regression with the haplotype at each marker. So here are our 20 chromosomes, 143,000 markers in the X bar, and each one give us an LOD value or the p-value. And we can see in chromosome 1 and chromosome X, there appear to be some peaks. That means in that region, there may be genes that are impacting the tumor onset age. And so the p-value, the log LOD value is pretty good here. It's over 8. So that means the chance of it being excellent is less than 10 to minus the 8th. So, okay, I hope I'm interpreting it correctly. So that is one readout. The second readout is the tumor growth rate. We have an equation that we derived that the natural log of the tumor volume actually is increasing proportionally over time. And the slope of the increased C1 is the tumor growth rate we use. There is a constant that we add to make the equation work. And so C1 is a single value. You do that, correlate that with 143,000 markers. And so the growth rate tells us the most interesting peak that looks most convincing is in chromosome 10. And so now we have 3 quantitative trait loci. Each one of these gene segment is called a QTL. So we have 3 QTLs in chromosome 1 and X and 10. So now we go to a series of steps. First of all, which genetic background is contributing to it? For onset age and chromosome 1, here is the LOD value plot in this one chromosome. And you can see there are different colors that spread out. So 0 is the average tumor onset, and red is when the tumor really happens earlier, so it's a negative value. And the red now is PWK, and next one is green is CAS. So here, if in this particular QTL, you have PWK or CAS, you're going to have early tumor. Same thing in chromosome X, it's also red and green. So verification, we were so surprised. Then if you look at the tumor growth rate, which is the slope or C1 value correlated with these markers. And here, this is the average growth rate, and the positive value is fast growing. So there you go. You have red and green again, and a little bit of WSB. So PWK and CAS, we predict, will bring about fast growing tumors. And of course, we're talking about these 3 genetic loci, but we can do a quick check, which wasn't so quick because we took bulb new T mice, which is the open circle. This is the spontaneous tumor, 14 to 20 some weeks. We crossed them with PWK. And we look at the F1 mice. They develop tumor between 9 and 14 weeks. So before your DO and bulb new T parent have the first tumor, all the bulb by PWK F1 mice have already got tumors. So it's truly accelerating in a very measurable manner. And so that gave us confidence that this is not a bunch of hoopla or witchcraft. It's actually mapping to something meaningful. And if you look at DO, it spreads throughout the whole gamut. And so now we got a little bit better with this one. The antibody titer study have already been reported, and this one is under review. And what we have a list of process that we go through to prioritize the gene. And very simply, if you do the single UCSD genome browser and look at all the genes in the region, the 3 loci have about 173 genes. And then you can do the SNP mapping. And it's probably easier if I just go there. So we know there are 173 genes. And we go to the Sanger genome, mouse genome database, and we ask which one of these genes have unique missense mutation or stop codon. And you're going to ask me, how about intron, non-coding region? We will get there when we could, but our first step is something that's a little easier to handle. So look at this gene. It has Cas, PWK, and WSB, and it's in chromosome 10. And chromosome 10 is the LOD, the peak is in, is from the tumor growth rate. So in tumor growth rate, the fast growth was Cas, PWK, and WSB. So this gene seems to fit the bill that it has unique changes in these 3 mouse strains. So, and this gene we'll come back to later. So when we do this in the 3 region, we find that 34 out of 173 genes have these unique changes. So well, our list has now narrowed down to 34 genes, but which one is relevant to people? I mean, I have cured mouse tumors many, many times, but so how many are relevant in humans? 26 of these genes have human homologs, and they are in similar 3 clusters. So it's interesting. And so which one of these 26 genes actually matter when it comes to human breast cancer? It may be important in other cancers, but for now we're focusing on breast cancer. And we work with CARES. This organization work with many medical institutions, including yours here in Louisiana, and they collect samples and they do whole transcriptome sequencing, and they have their own in-house algorithm called Code AI that link the clinical data with the genomic data. And once you establish a protocol with them, they will assign scientists to analyze your data with you, and you can pose different kind of questions and bring out the information you need to know. So we look at 35,000 patients with primary breast cancer and about 5,000 patients that have metastatic breast cancer. And we ask if any one of these 26 genes makes a difference in the breast cancer outcome. So here's the primary and here's the metastatic patient population. Red is the one that has high expression of LEORB4, blue is the low, and you can see high expression of this gene results in shorter disease-free survival, or shorter overall survival. And the p-value is extremely high. So there are genes, and how many genes? So here is our list I will share with you. Mouse chromosome 1 is human chromosome 6, 10 is 6, and 6X is X. And they are all same clusters as in mice, with one exception. LEORB4 is now in chromosome 19. And here are the 26 genes, and 21 of them are relevant to breast cancer outcome. So that's 80% of the genes we identify with mice have human clinical correlate, saying they actually impact breast cancer outcome. And what kind of genes are there? GLE2 is almost strictly in the stroma, only in the tumor fibroblast or normal mammary gland fibroblast. DBI and SCTR are in lipid metabolism. So it's really kind of an interesting list. And there are, of course, the ROS gene, which is in the mammary tumor. So which one of these 21 genes should we start? All of them are interesting, and all of them are probably important. So to get started, we said, do they impact the same pathway? And so we just did a quick pathway analysis. So 12 out of 21 genes share some kind of pathway. And what stood out to us is TSC22D3 and LEORB4, because they actually show direct interaction. And what's TSC22D3? It's actually a transcription factor. It just makes a complex with glucocorticoid receptor, and the complex sits on the promoter of LEORB4. And when LEORB4 is turned on, all kinds of ligands are in the mammary gland environment made by the tumor, by the stromal cells. When that is turned on through signaling, they actually start to make arginase, the NOS, and various cytokines. And we just heard that in the last talk. It's fabulous that, okay, maybe this is important. And we didn't pick this out of the blue. We picked this out of a real live growing tumor. And the tumor is telling us that LEORB4 is what makes my tumor early or my tumor late. And so we just feel this is approaching things from the system itself and maybe could be relevant. So we're excited, and I'm excited to hear about the macrophage story, that LEORB4 may be a possible molecule to look at. And recent literature, from Jim Allison's lab, for example, is describing LEORB4 as a myeloid cell checkpoint molecule. And there's effort now to target this molecule and block it perhaps during immunotherapy or to combine with any other spectrum of molecules. And it takes us out of just the T cell, out of just the B cells, but into the antigen presentation or into other functions in the immune system. And so where do they express? So you just a quick simple cell RNA data, which you already have. So you just go back and look. LILRB4 is found in normal mammary gland, tumor adjacent normal tissue, somewhat quasi-normal tissue, and in the tumor. And the prominent expression are always in the macrophages. And we don't do the wonderful proteomics. I have to talk to Ellen afterwards, but we just do flow to verify. And sure enough, they are expressed by F480 positive macrophages. So we feel that this is a good point to sit down and say, what experiment can be done? As we speak, we have processed the MTA and the LILRB4 knockout mice are coming our way. And first thing is we're going to make our own antibody. We can just do DNA electroporation, man, we know how to do that. So you know, many possibilities, and LILRB4 is just one of 21 genes, right? So there are other possibilities, but for tumor immunologists, this stands screaming at you. And so here from this second QTL analysis, we believe that the tumor progression is regulated by many heritable factors. And again, the DOF1 platform is very effective in identifying these regulatory genes. And we have at least LILRB4 on our panel that is actionable and possibly a very plausible target for immunotherapy. So these are the amazing people who have worked with us over time. Jane Jacob is a junior faculty in my group. She just submitted her own R1, and she did a lot of the study, especially in the second part of the talk. And I want to thank all these amazing people and the patients who participated in the trial to make it possible to advance. Thank you, and I will take any questions.
Maybe I missed this, but can you modulate LILRB4 in, is it differentially expressed in non-stimulated or myeloid cell that comes fresh out of bone marrow, and after it becomes part of that tumor microenvironment? Maybe I missed it.
You did not miss it because I did not say.
Can you please repeat the question for the remote audience? Thank you.
No, repeat the question. People can
Oh,
hear.
I'm so sorry.
Thank you.
Okay, the question is a very smart question. That is, does LILRB4 level varies when it matures out of the bone marrow versus after it gets into the tumor? I don't have a good answer to it. In fact, I'm just looking at this single cell data. It doesn't look that different. In fact, it seems to be, you know, this, of course, this is relative level, but it doesn't look that different, but actually seems to be a little lower in the tumor. But we shall find out. In fact, I should talk to Ellen Tuckett very soon about this.
We have a question from the remote audience. Dr. Garden.
Okay.
Thank you very much. What an awesome talk. Thank you so much. My goodness, that's impressive. Really beautiful, beautiful marriage of genetics and immunology.
Thank you.
One of the things that we're now increasingly aware of is the effect of the gut microbiome and the response to tumor vaccines. Admittedly, all these mice were kept in the same environment, so you'd imagine that they might have the same gut microbiota. But I wonder if that's something to consider too, vis-a-vis difference in genetic makeup, and often that has an impact on your gut microbiome, and that may subsequently have an impact on responses to vaccines.
You know, I would definitely entertain the concept, and for this kind of study, as I said, you want as little human intervention, interference as possible, and change in diet is #1 on our list, because you know that would change a lot of things, and you'll have easy readout and you don't have to manipulate the mice. And microbiome has been implied in so many ways. There's really no reason not to. It's just that, and we will have it on our table. We just need to breed 5 to 1,000 mice in order to get enough for one experiment. But once you have it, it's worthwhile. Look, you just find Leo RP4. It's worthwhile. Yes.
Thank you.
Thank you. Thank you so much.
Interesting analysis. There's an area of high AO score. Are they going to target the area instead of single gene?
Well, the question is, the QTL covers a range. You know, it has a range of genes in there, and we are picking out single genes. How about other genes? Is that what your question? Or should we target the entire region somehow? Yes, so the region that has high LOD has protein coding genes and also has non-protein coding genes, right? And the genes that's non-coding may actually be even more interesting. Sometimes it's distant promoters, sometimes there are various mechanisms that will impact multiple genes. And we are now just starting to work with a geneticist who look at this methylation, acetylation, et cetera, to see if we can find a way to handle it. It's not as easy as working with genes that code protein, and you can know, you know, if you have missense, you have stop codon, then you will change. But those non-coding genes, non-coding regions are a bigger challenge. And if you have expertise here, we would love to work with you. Because you know, information is out. You can look at that region yourself to see if there are regulatory region that you believe would be of interest, and we would certainly be happy to work with you on that. Thank you.
I got a question about the level of tissue types, because we mentioned that it's always expressed in council and that we can what if I think we do for tools specifically in response is too strong and these spots are attacking normal for this question. So that's my first question. The other question is to the experience of IgG animals. Did you see allergy specific?
Okay, the first question is the HER2 expression profile. If you have a strong immune response, are you going to have a toxicity issue? And the second question is you want to know about IgG in B6 mice, and you want to know if there are T cell response.
Yes, I was wondering whether or not it was just a type of immune response.
Right. Okay, good. I'll answer the second one. In B6 mice, there's hardly any T cell response. So the antibody and T cell response actually go hand in hand. When you have more T cell or more antibody, you usually have more T cells. This is true with HER2. You know, it may not be true with the other genes, but with HER2, that is true. Now, in terms of HER2, yes, HER2 is expressed in our heart. And that has been a toxicity in the Herceptin trials. And so now you have to be very careful when you treat with patients that they don't have a heart condition that may become a side effect that would be out of control. Of course, in those treatment situations, you give very, very high dose HER2 antibody and for long periods of time. For vaccination, we're proud of what we can induce, but it's a far cry from what you would give with monoclonal antibody therapy. And so I think it's something we should be vigilant about, but it may not be a huge issue. Thank you.
Thank you.
Well, thank you very much.